Well, hello. Well, this is our group, Andrea LayedraA and Roberto Leguizamon, my name, and we are going to talk about Staphylococcus aureus, specifically methicillin resistant. Next one.
Okay. The main contents of these presentations are microbiological characteristics, infection mechanisms and pathologies, and a matter of concern that includes mechanisms of developing resistance, high-level resistance to methicillin. Regarding microbiological characteristics, Staphylococcus aureus belongs to Staphylococcaceae family, genus Staphylococcus, and species Staphylococcus aureus. Is a gram-positive bacteria, single in pairs or short change of 3 or 2 bacteria. And its diameter is 0.5 and 1.5 micrometers. It presents a cell wall very thick of peptidoglycan. It's non-flagellated and not motile and non-sporing bacteria is encapsulated, and it forms grapes like cluster arrangement. And regarding the habitat of these bacteria, it is present in mammalian body surface, in normal flora of skin and mucous membranes, and present in nose, specifically in anterior nerves. Regarding cultural characteristics, these bacteria grow on most bacteriologic media under aerobic or microphilic conditions. Its colonies are round, smooth, raised, and glistening, and forms gray to deep golden yellow colonies. To identify these bacteria, the following agar media are used. Manitosal agar, in which the bacteria appear circular, of 2 or 3 with smooth and shiny surface colonies, and it appears opaque and are often pigment golden yellow. In triptych soy agar, they appear circular, convex, and entire margin. In blood agar, shows beta hemolysis, and in brain heart infusion agar, form yellow pigment colonies. Regarding virulence factor, these bacteria express many potential virulence factors that include surface protein that promote colonization of host tissues in vaccines, surface factor that inhibit phagocytic engulfment, biochemical properties that enhance their survival and phagocytes, immunological diseases, a membrane damaging toxins that lyse eukaryotic cell membranes, exotoxins that damage host tissues and provoke symptoms of diseases, and inherent and acquired resistance to antimicrobial agents. The mechanism of infection, as you can see in this figure, the bacteria obtain access to sterile tissues via open wounds and use adhesive proteins such as fibronectin binding proteins, clumping factors, and collagen adhesins for a specific attachment to extracellular matrix protein. Staphylococcus aureus can also in part regulate polymorphonuclear leukocytes influx in some ways that involves activators and inhibitors. These bacteria produce coagulates to polymerize fibrin and form an encapsulated abscess around the infection site. And these bacteria can comprise effective opsonizations by antibodies using the polysaccharide microencapsulate and surface protein that binding immunoglobulin G. And the abscess can release a live bacteria to the surface of the skin and or the bloodstream at later stage. And the pathologies, human staphylococcal infections are frequent, but usually remain localized and the portal of entry by the normal host defenses. This portal may be a hair follicle or respiratory tract. And the localized host response to staphylococcal infection is inflammations characterized by an elevated temperature at the site, swelling, the accumulation of pus, and necrosis of tissues. A resulting septicemia may be rapid fatal. Bacteremia may result in seeding other internal abscesses, other skin lesions or infections in the lung, kidney, heart, skeletal muscles, or meninges.
Okay, now talking about prevalent clones. Next one, please. It has been seen several clones around the world because it's very well distributed, and we are going to talk about the specific ones in each continent. For example, in South America, the prevalent clones are USA 1100 and USA 300 LA, Latin America variant. Then that's CAMRSA, that stands for Community Associated methicillin-resistant Staphylococcus aureus. Those ones are the predominant in South America. In North America, it's USA 300, the sequence type A4, that's also a CA. In Europe, the predominant one is sequence type 84. In Asia and Africa, because of some reason, it's not well determined which is the one prevalent, the prevalent one. Yes. Next one, please. Something to mention also is other clones that are that must be mentioned are these EMRSA 15 and 16. These clones are part of the epidemic methicillin-resistant Staphylococcus aureus, that because it stands for these siglas, lineage prevalent in the United Kingdom. And then we have sequence type 239, that's HA, stands for healthcare associated methicillin-resistant Staphylococcus aureus, and this clone is one of the most, it's the one multi-drug resistant, the most widespread in the world, that's present in the whole world. And one thing to say is that these HA are more aggressive in terms of resistant to antibiotics, because they are more resistant to more antibiotics than the CA. Next one, please. How we treat. So, of course, it will depend on not only the susceptibility test, the antibiotic susceptibility test, but also where is it and how is it this infection. We have three focus that we have to focus in the treatment. Next one, please. Now going specific to the therapy. So how once it's done, because of course there is not the only one specific treatment for all of the Staphylococcus. It will depend always in the antibiotic susceptibility test, but there are many antibiotics, as you can see here in this figure. It will depend, of course, in the target. Where is the target, that will have an antibiotic. The most that in the practice in the clinic are used vancomycin, daptomycin, linezolid. For example, those three ones are the most used. And well, here says existing antibiotics and options are in green, new antibiotics approved and on the market are in blue, antibiotics in the pipeline are in orange. Yes. Next one, please. And well, talking about the other points, the site of infection and the severity of the infection, we have this incision and drainage, wound care, and supporting measures. So because this procedure of incision and drainage, this procedure involves making an incision and drain the pus. Wound care, of course, yes, this may involve cleaning the affected area, for example. And supporting measures, yeah, therapy could be another, but because in severe cases where the infection has been spread, so we need supportive therapy. Next one, please. Mechanism of development resistance. How this resistance is developed. Well, we have many, as we have seen it during the course. One is this modification is the PBP, the penicillin binding protein, the affinity. So because it changed the affinity, the antibiotic cannot bind. So it is not an effect, and the microorganism can grow even though in the presence of an antibiotic. And primary is attributed to acquisition to the MEC-A or MEC-C gene, and this gene encodes for this PBP. There are 12 known staphylococcal cassette chromosome MEC types from 1 to 12 have been identified and are classified according to the type of cassette chromosome recombinase complex and the class of MEC complex. Next one, please. PBP has a reduced affinity for beta-lactam antibiotics, as I said, including methicillin and other penicillins, which allows the bacteria to survive and grow in presence of these drugs. The SCCC-MEC element carries additional genes, including regulatory elements that control the expression of MEC-A, for example. Next one, please. Beta-lactams production. Yes, so there is not only these strains can produce these modified PBPs, but also produce beta-lactamases. So, and these beta-lactamases can further degrade beta-lactam antibiotics and can contribute to the resistance. So we have both mechanisms acting together. Not in all of the MRSA, but it could happen potentially. Next one, please. It's important to know that the mechanism of resistance can vary among different strains. And this is two cases very important to say. Vancomycin resistant in Japan, and here in the Hospital de Clínicas, there was a case with linezolid resistant Staphylococcus aureus. So during the treatment, this microorganism developed resistance against those antibiotics, which is really worrying, but as expected, because you know when you introduce an antibiotic, an antibiotic, the microorganism will try to avoid that and grow. This is what happened here. Next one, please. Mother or concern, high level resistance to methicillin. Yes, MRSA epidemiology is changing rapidly due to continuous development of clones, deficient therapeutic management of infectious diseases through automedication, the lowly restricted sales of antibiotics in pharmacies, as well as medical prescribing practices without a laboratory investigation contribute to the development of new MRSA clones with increased resistance to antibiotics. And because of this, multi-drug resistance phenotypes are emerging. Next one, the knowledge of clones allow studying antibiotic resistance spectrum for each one, a fact of great importance for medical practice. Appropriate prevention, risk factors, natural history of staphylococcal infection are not fully elucidated. Therefore, it's necessary to monitor antibiotic resistance, judicious organization of antibiotics prescription and therapy of infection contagious disease, medical education of travelers and immigrants. Next one, here our bibliography, and thank you for your attention. Yes.